Welcome, and I don't know if I'm pronouncing this correctly, but Heloisa Bordallo, is that? No, that's not at all correct. Is it? Where?
Heloisa Bordallo.
Where's this from?
I'm from Brazil.
From Brazil. Okay, great. Anyway, yes. So the topic of today is how to integrate machine learning into physics experimental pipeline in the context of vast amounts of data, and how you can maybe use machine learning online to make decisions about this data. So I'm very much looking forward to it. And I should mention that you're from the Niels Bohr Institute.
Great.
All right, thank you very much.
Great. Thank you for the invitation, and thank you for being here, because basically my talk is like I'm asking for help. And I am here to explain to you the problem we have. We have people here from the Niels Bohr, people here from the Data Management Center, from the European Spallation Source, and we are reaching out for help. So basically, to get help, I think the first thing is that we have to understand each other's language. So forgive me, because really, for me, AI, deep learning, and machine learning, it's all the same. Okay? Because I have to learn it. I don't know it. So my idea for today's talk is to tell you what neutron scattering is, and X-ray scattering, tell you what we do with it, what we could do better, and then from there start some kind of discussion. So I have a double hat. I am associate professor here, but I am also part of the ESS because I was one of the main proposers for an instrument called Miracles. And yes, indeed, we can do miracles. However, miracles takes a long time to happen. This instrument is going to be one of the last ones at the European Spallation Source. As you can see here, it is not a very cheap machine. It's a lot of investment, a lot of people. And what we basically do with this type of instrument is that we measure dynamics in different time scales. So what I mean by that is that, for example, if we take a water molecule and we have in our body a lot of water, right? In different parts of our body itself, we have water moving at different time scales. And a lot of diseases can be diagnosed if we look how this water is diffusing. So neutron scattering is yet another technique that we can use to look at water diffusion, just in a very, very basic point, and then we are going to look at it in more detail during my talk. However, even if we are at the state of the art with instrumentation, we still do analysis as we did in the 80s. So this here, it's not that it's a very bad resolution picture. It's because the paper is not very new. It's from '92, and the resolution when it was put in PDF is very bad. And in this paper, what it is said is that quasi-elastic neutron scattering has a lot of potential in '92. We need better instrumentation. What I want to argue is that the very good instrumentation is already there. So this data here was taken, the same instrument that this data was taken in 81 and in 2019. We analyze the data still in the same way. Why do we do that? Because quasi-elastic neutron scattering is a broadening of elastic line. So here I have intensity and energy. I have energy equals 0, and then I have a broadening. And this broadening tells me about relaxations. So that is why I can figure out what is the water diffusion in different systems. Okay? So we analyze it by deconvoluting Lorentzians, as we always did. However, we do the same thing, doesn't matter which system we are studying. Because with this technique, we can use to analyze water in clay systems, we can analyze water in proteins, we can analyze water when it's hydrating cement for dental cement, for example. We can also use this technique to see how a drug interacts with a cell and how the water dynamics change. So we have a hammer for all problems that we are looking at. So that's basically, again, what we do. That's the signal I have. I will say, okay, if I have something fast, I have a broad Lorentzian. If I have a motion that is slow, I have a narrow Lorentzian. Okay? So if I just go back here in this slide, what the question is now, with the ESS instruments, we are going to surpass the amount of data that we take by a lot. And what do we do with analysis? Well, that was a master thesis of this student of mine. What we did during his thesis is that we wrote a new program to fit my data with Lorentzians. Okay? The same, over again. And then what we get when we have these Lorentzians is that I have a line width. So I will plot this line width as a function of the scattering vector that I will explain to you a few slides later on. And then here I have a shape, and depending on the shape, I will say I have either a free diffusion, I have a diffusion inside of a sphere, I figure out something, a hand wave. Okay? That's what I do. But then, if I really start digging, machine learning, because I know a lot of systems, and if I look at papers, I will see that when I observe this line width as a function of this scattering vector to the square, doesn't matter if I'm looking how water dynamics change when it's hydrating on cement, or how these water dynamics change when I have a protein with and without an inhibitor. It's the same. The only difference here is that this is plotted in milli-eV and this is in micro-eV. But these data are identical. Don't worry. Later on, I will tell you that with a little bit more thinking, we can explain what we are looking at. But what really scares me is that we use the same analytical model, same simple model, to analyze all these different systems. So what do we do really in a quasi-elastic neutron scattering experiment or in a general scattering experiment? What we have here, we have a neutron that will come with an incident energy or a state that I know. It will interact with the sample, and then we have the scattering process, and it will be detected. So it's very simple. And then, depending on what happened, if I have an exchange in energy, I can measure this exchange in energy. If I have a diffraction experiment, so I'm looking at structure, I don't have exchange in energy, I'm doing an elastic experiment. The interesting thing about neutrons in relation to any other technique is that we can also have information on the scattering vector that I was talking to you about, that is this Q vector here. And having information in this scattering vector not only is going to tell me if the particle is moving, but it's also going to tell me about the nearest neighbors. So that is why, for example, in our study with the drug molecules and cancer cells, I can figure out if something is happening because the drug molecule is interacting with a water molecule or with the cell itself, because I can have information on the nearest neighbors. And then if you ask me, okay, what is then the difference between X-rays and neutrons? It's because with X-rays, what you have an interaction that will vary with the electronic shells. So if you have something that is very heavy, have a lot of electrons, I will have a lot of intensity. If I have hydrogen in my sample, I have a problem because I don't have a lot of electrons, okay? With neutrons, this interaction is different because it's an interaction with the nuclei. How strong is this interaction? We know because we have measurements on these cross-sections. So what happened here is that while for X-rays, hydrogen, we can say that is basically transparent, for neutrons I can have information on that. And that's why I told you that we can have information on the water dynamics, because I have my scattering vector and I also have this very high cross-section. And then, for example, if we are looking at magnetic systems, it's also very important, because when we look at the periodic table, you're going to see that manganese and iron are very close in the periodic table by the number of electrons. However, the cross-section for neutrons is different. So by doing that, we can have a lot of differentiation in materials. The other thing is also that we have this interaction here with the nuclei. So if we use a deuteration, I can hide or highlight things because we don't have the same cross-section between hydrogen and deuterium. So then here, if we put that in an image, what means is that if I do an X-ray hagiography, what I am going to look better is the metallic part in all the systems that I'm giving to you information here. While when we have the neutron hagiography, what we are going to see is the lighter elements, right? And the other thing also that is quite interesting is that, for example, if I look with my naked eye in this picture here, I cannot really see that I have something hidden behind this metallic part that is here, because it's something in carbon and I have a different contrast. Then I'm talking to you about cross-sections, what that really means, right? When I do a measurement, at the end of my measurement in the detector, what I'm going to collect are the neutrons per second. This number of neutrons per second that I collect, it depends on the sample, that's the number of scattering elements in the volume, and also in the cross-section, here, the scattering cross-section. So what also makes interesting doing neutron scattering is that I can quantify what I have in the beam, because the intensity is directly proportional to the amount of material and to the cross-section and to the density of that thing that I'm measuring. Then the other things, of course, this intensity will be a relation with the neutron flux, the solid angle, and so on and so forth, and that is why we develop better instruments, because I want more intensity. Then, what does it mean again if I translate it in another way? What we measure here is that if I have something that is structured, so I have a structure where I have elements that are repeated periodically, what we have here is that I can find the same element in another position. So this will give rise to me to a Bragg reflection, and that is what I measure when I'm doing a diffraction experiment. And that is directly proportional to what we call the coherent cross-section. Now, if I have some kind of system where I don't have this periodicity, I don't have this arrangement, it can be a liquid or it can be a polymer or it can be a structure that is disordered, I lose this coherence. So what I measure then, it's something that can either be a background or a continuum. And because of that, we call that incoherent. But these are static measurements. Then what happens if these things start rattling around the equilibrium position? Then can either rattle in a coherent way, so if this atom moves, the next one has to move together because we are still ordered in these lattice, right? So we have to be coherent. And then what we're going to see here is what we called an inelastic vibration, because I'm observing a change in energy, but not at energy close to zero, like my problems with quasi-elastic neutron scattering. And then this again is coherent, but now we talk about double differential cross-section, because I have a change in energy and in position. And then if these things start moving in a way that is completely incoherent, so it rattles around, that's when we observe the quasi-elastic signal, that is this broadening of the elastic line, and again, that is a double differential cross-section and is related to the incoherent cross-section. Okay? So that leads us to describe neutron scattering with basically two different types of instruments. I either have diffractometers or I have spectrometers. With diffractometers, what I'm going to do is that I'm going to measure structures. Small structures, crystalline structures, or very large structures like protein structures. And then the only thing that we really need to remember here is that I need to know Bragg's law, that is basically interference, constructive interference, and I don't have an exchange in energy. Or I will use spectrometers where I'm going to measure dynamics, and what is valid here is Newton's law. And as you can see here, our instruments are very big because neutrons are very difficult to produce and very difficult to detect. But still, compared to particle physics instruments, they are not that big or that expensive. So first thing that we are going to do, we are going to look at diffractometers. So what we can do with diffractometers is that we are going to measure length scales. And we are going to do experiments that can give us information from the nuclear particles up to radiography and tomography for real big stuff. We can take, for example, a full motor of a car or a turbine of an airplane, put it in a neutron beam line, and analyze it. And then here, what I'm going to show to you, it's a typical neutron scattering experiment. And what we are looking is angle and intensity. And this is what we call the Bragg reflections. So we have some structure, right? We have this periodicity. So we have elements that are going to repeat. And because they repeat, there is a constructive interference. And then we observe these reflections. We can analyze this data using Bragg's law and some other programs that we have to analyze, and we come up with a structure. What becomes then interesting is when we have a phase transition. So, for example, when we have a system that is what we call paramagnetic and becomes magnetic, because magnetic systems are important for different applications. It is also interesting for me when I have something that is liquid and becomes solid. So that is what I'm looking at. So these are changes that are going to be detected when I'm modifying the temperature of the sample. So this is basically an online data acquisition, and what we do here is that we have the sample in one of these big diffractometers, and we are going to change temperature. And then what we have to concentrate, so this is the whole pattern that we can observe. So again, intensity and angle, and we are only going to concentrate here. Because what happens when I have a phase change is that I will not have the same pattern. So I can have a minuscule splitting of a Bragg peak. And that's what we are looking for. So if you look at the movie that I'm going to play for you, you're going to see that there is a certain point where I go from this double peak here to a single one, and then it doubles again, because it's also what we call a reversible phase transition. So then we are changing temperature. The sample is going to another state. Now we have one single peak, and then if we go back and change temperature backwards, I go back to my same structure. So this is a typical experiment, and it's something that we would like to be able to predict. Can I, during my data acquisition, have something smart that comes from some machine learning and tells me, well, wait a second, the way that you are putting your experiment to run is not really the correct one, because that subtle change you are missing, because you are going too fast or you are going too slow. So can we control already my instrument when we are taking data? Then another way of doing diffraction is then when we do small angle neutron scattering or small angle X-ray scattering, small angle scattering. Don't tell the people that do small angle scattering that I call what they do, they measure blobs, right? They measure things that are big. So, but still, Bragg's law is valid here. It's diffraction. It's just that instead of Bragg's law, we are going to a small angle, so we have this approximation. And then what they have here is that they will measure what we call a structure factor and then a form factor. And from analyzing this form factor, they can tell what is the shape of this blob. Is this blob elongated? Is more like this, or do we have a core and some hairy things? And also we can look at some magnetic structures. So when we put in a map together, what we can do with elastic neutron scattering, there is not one single measurement. There are many different problems that we can look at. That is great, but at the same time it's a curse, because we do not have really only one single problem. So what is happening now, this comes from a workshop that was at the ILL last year, and that is something that is being developed at EMBL and in Hamburg. And what these guys did is that they used some machine learning algorithms. I wasn't in the meeting, so John told all that to me, but this is also online and you can go and listen to these talks. So what they did is that they used some machine learning algorithms to predict the shape of the curve they would measure. And they compare that with a huge data bank that is available, because small angle scattering are very fast experiments. And there are instruments that are extremely good that you can also do in-house experiments. So it is very easy to compare existing data to prediction data, right? So that is what is going on in the side of machine learning and the small angle neutron scattering. So now, if we move to the spectrometers and if we move to dynamics, these are the time scales that we can look at. So we can go from spectroscopy where we really measure very small interactions to things that are, again, time-resolved hagiography, where we can see, for example, a lithium battery at work, and we can figure out what's the diffusion of that lithium battery. So when we are doing this type of experiments, things become a little bit more complicated because I put another parameter. I put time into play. And because I put time into play and I'm looking at dynamics, I am really opening the umbrella of problems that I have. So when I do this type of experiments, remember, we have the coherent cross-section, so we can measure phonons, that they are collective motions. So things are really moving like a marching band, or I have single particle diffusion, that you can imagine that's what happens when you are in a dancing floor. I don't care, I just move. And because of that, we are able to measure all this energy transfer range, or that can be translated to time scale, and that can also be translated to the length scale that I'm probing. So again, we have information in many different types of samples or materials. So now I'm going to show to you a movie of how this type of instruments run, so that you can understand a little bit of the complexity of the measurements that we have as well. Because when we have this type of spectrometers, not only I can have these different materials that I can have information from, but I can also configure my instrument in many different ways. So hopefully the movie will work. So this is what is the IM5 instrument that I showed to you data from the beginning. And don't ask me what these arrows are. I couldn't take them out from the photograph on the web. So the reactor is here, where the neutrons are produced. We have what we call a neutron guide hole. This is the spectrometer. This, I think, is Jacques. That is the person responsible for the instrument. We mount the sample here, and here is the bank of detectors. And then the guide continues to another instrument. We have many instruments after the other. So here, neutrons are produced in all directions with all energies. We have then to use a chopper system to deselect a particular energy that I want. Then the neutrons, when they interact with the sample, again, they are scattered in all different directions with different energies, and then they hit the detector. And then when they hit the detector, I do some manipulation, I reduce my data, and what I get at the end is what we call here a spectrograph, where we have intensities, and then we have energy. So just for you to remember that when I'm doing an experiment, I have my idea, right? As a user, I have my preconceived idea. I'm going to measure some excitation at this energy. So what I do, I tell the instrument scientist, great, configure for me the instrument on this way. Run the choppers in this particular frequency so that we can select a particular incident energy, then I will have my data. Well, it's not that easy, is it? Because I can have many different energies. I can have many different energies that I scattered. So how sure am I that what I really want, my discovery, I missed it because I either didn't know how to configure the instrument, I either didn't reduce my data correctly, well, then the analysis is another issue. So that again, can we use machine learning to help figuring out how to really take the best of these instruments, how to configure this machine, how to take out my preconception. And again, when we have, I'm going to show to you later, when we have data, we are covering vast amounts of energy and momentum transfer. Maybe my data is under the noise somewhere else that I didn't look because I'm not trained to it. It's my eyes, like machine learning here is a little bit limited. So again, when we're talking about this type of instruments, these are all the machines I can choose from. And basically what I do is that, okay, I am doing some lattice vibrations. This is the type of instruments I should go for. I look at the list of proposals, and that's how I choose. So modeling inelastic neutron scattering data, this is at the end some of type of data that I have. This is my data. I have here intensity and I have here energy. And this is paracetamol. Okay? Paracetamol that we take for headaches or whatever. Paracetamol can crystallize in two different structures. And that is why my spectra is different, because the structure of this drug is different. Monoclinic is the one that we can buy in the pharmacies. Orthorhombic is the one that people in the industry would love to have because it will make production cheaper. However, we don't know how to make this stable. So what I found in the literature from a group in Germany, is that they are using some machine learning, so gradient domain machine learning, to produce spectra that will reproduce what I can measure. And I will go back to this figure a little later to show to you what is my state of the art and how fantastic it would be to be able to use this type of approach to explain this type of data that we measure in facilities. Oops. Okay. So then I just want to put a step back to tell you a little bit of what I do and tell you why I'm asking for help. So that is the periodic table, but I am a little bit simplistic. So my periodic table starts and stops at hydrogen. And this is the type of research that I do in my group on the other side. We go from dental cements to cells, to vaccines, to antipsychotic drugs, name it. If it has hydrogen, it's fantastic. We can try to do something. So in the case of the antigen load nanoparticles with vaccines, what was our interest here is that we don't want needles. We want to be able to deliver vaccines or drugs with modified delivery systems. But to do that, we need to be able to understand how a drug interacts when it's confined somehow and what is really happening in this confinement. Neutron scattering is very good because neutron scattering can deeply penetrate in a sample. And neutron scattering is also very good because I can see the hydrogen. So to answer this very simple question here that was asked to me by some epidemiologists, we have just to do these very many experiments, some of them in the lab, others in large-scale facilities. What was very good about these results is that when we went and we did imaging, X-ray tomography and neutron tomography, that's what I'm showing to you here, on the samples that we had. So these are powder samples, and what we have is that we have this antigen loaded in this sample. And the question that we had to answer for the people that are doing the tests is that they knew that depending on the amount of antigen, we have a lower or higher biological response. And why was that? So by combining these two techniques, what we could tell them is that what happens here is that the antigen is put together with a salt, it agglomerates, and this agglomeration is not uniformly distributed. And what we can see here is that with X-rays, we can locate the salt because I have heavier elements, while with neutrons, I can locate where the protein is. And what was very surprising for us in this type of experiment is that if I mix it around, it will be very difficult for you to tell me which sample is which. But that is exactly the conclusion that we have. We have agglomeration of the protein with salt. So it would be very nice to be able to have some software that helps analyzing this data. Because with the student I had, although he was brilliant, I am sure that we have much more information to dig than we could really have. In the case of dental cements, what we want to understand is we want to understand hydration, because you know that sometimes your filling from your caries cracks, and it cracks because, again, you have liquid diffusion, and you have a porous system, and because you have this diffusion, the material cracks. Our filling cracks in the same way that the walls do, and all because we have this dam water that is moving. So here again, what we did is that we combined conventional dental X-rays to X-ray tomography and neutron tomography. And again, this is the beautiful feeling that a dentist will see. This here is what we see with the very large pores, and with neutrons what we can observe is that some of these pores are filled with liquid. Being porous is okay. We need porous materials because they can take stress. This is not okay, because if we have these large pores filled with liquid, this liquid will diffuse and we will have cracks. So the question that we really have here, together with my collaborator in odontology, is that could we somehow, in a much clever way than what we're trying to do, is to figure out that we can develop better materials. Can we basically have some prediction of the hydration process and how we can control it? So we're using quasi-elastic neutron scattering. Here it's my student Marcela. She went all around the world to do these experiments because we want to understand how this liquid was diffusing. What she came up with was one of the figures that I showed to you. So that was the broadening that she got. And then here we have that famous same figure. And what we see is that during her 6 months change of environment, she went to the US. and she was able to do molecular dynamics simulation in a superfaster computer. So what she figured out here is that in these materials, what happens is that we have the polymeric chains, because the liquid that we mix with the glass is a polymer. And then these polymeric chains, we will have some water that is trapped here, some water that is closely bound to the polymer, and some water that behaves bulk. So this behavior that she got from the analysis of this line width tells us that what we have is that we have water molecules diffusing in different landscapes. So we have a water molecule that, of course, is not just static in this environment, but it can move around. This is very expensive because she could do that only for one sample. It was 6 months that she had to stay there with a lot of money in supercomputing. And then now we have a new project together also with the data management, that the idea is to use some molecular dynamics software, but easier one, because one other thing is that not all users that come to the facilities are expert physicists. We have people that come, as I showed, from epidemiology, anthropology, biology. So we need some tools that can be black box for biologists, not for physicists. So we need also to make things accessible. We need to make somehow molecular dynamics correct, but accessible. So now I go a few steps back. And that's what I was mentioned to you, right? How to monitor that what we are collecting is the right data set. Because as I mentioned, I have infinite ways to tune my instrument. We will not, because no instrument scientist will try something new because it's very risky. And as a user, I will also not, because I have one shot to do an experiment and be successful. But we still need to think in ways that we could do targeted data acquisition. So this is some results that came out from a workshop that we held last year at the Newsborn Institute. And the thing is that one point that was discussed, can we have some adaptive algorithm use machine learning to make some decisions? So I go back to this slide, and I think this is a very good example. Because if I have somehow predict the shape of my curve, and after 5 minutes I already have it, I can move on. Because we also like to measure very much with very small error bars, and maybe that's not needed. So it's really taking decision on the fly. Yes, that's what you have, correct, next sample. That's not at all what you have. So in this case, think. Maybe your sample degraded, maybe you're having agglomeration, maybe your system is not at all what you have. So I need a red flag, that's not my user eyes, right? So can we do something in this direction? And then the other thing is that, the other point, if we go another step back, how can I monitor that I am doing the right data analysis? How can I monitor that I'm doing the data analysis correct? So that is what I mentioned, that we have these thousands of individual experiments, and the data volumes are becoming larger and larger because our detectors are becoming larger and larger. We are producing more and more neutrons per second, and we are producing more and more photons per second. The non-expert users, and then basically what happens, a lot of data is quickly analyzed, but not fully analyzed. So there is a lot of things there that are just there. And then we don't have any routine in any beamline. So it's really like a field that can be fully explored. And then the thing, yes, is that worth to look at the promises of machine learning and deep learning in order to extract the data that we have, or in order to get us to look at the data different, in order to get us to think how to do measurement different. So there are opportunities for doing better. And I go back to my example on the paracetamol. What I'm showing to you here is my experimental data. So we have, again, the monoclinic and the orthorhombic sample. This is intensity, this is energy. This is the state-of-the-art GFT calculation I can get. Why I don't plot them together? Well, because they really don't fit, okay? They don't fit. Then if I go back to what I found in the literature, and I look what they got, and I compare by eye with my experiment, it's damn good. It's much better than what I can do now. And then of course, you're going to tell me, no, it's not that good. The thing is that this is a very simple calculation, as they explain in the paper. To get to my data, there is only one next step, because we need to convolute what we call the resolution function. And we need to transform that in S of Q and omega. There is one simple step here. And I guarantee to you that this, already even without any step, is much better than what I can get. That would be a huge improvement, huge improvement. Then I show to you another problem I have, is these porous organic polymers, right? I have no structure. To do this kind of experiments that I show to you, to have the GFT, I have to have a periodic structure. I don't have it here. The interest in these little things here is that they can contain ammonia. So they are very good for cleaning the environment or to being used as masks. But I have no clue what the structure is. So with my very clever chemist colleague, we can propose structures. And then with these proposed structures, we can do state-of-the-art GFT calculations. This is machine learning, but machine learning brute force, because I have an extremely good collaborator, and what he did with his patient is that he took these molecules, he turned it around, and he mounted the system like that, and voila, we got these very beautiful calculations. And I tell you, it's much better than what is out there. That is state of the art. It's ridiculous, but it is state of the art. It's top. So the thing is that if we can apply some machine learning algorithms, and we have to think that in most systems that are interesting for us, we don't have local symmetry, so I cannot do simple GFT. I have global symmetries, and global symmetries are really where the interesting systems are. So there is some hope. Then there are other opportunities also for doing better. This is a beautiful set of data from IN5 in what we call molecular magnets. Molecular magnets are very beautiful and very simple systems because they are one dimension. By being one dimension, it is really possible to explain this data measuring, assuming something in a theory that we know, fitting the parameters, and getting the spectra. It's fantastic work that this Italian group does. It's really fantastic. It's one dimension. But then some things are not one dimension. Some things are much more complicated. And that's also very interesting here, because these systems can be used as quantum dot systems, these systems also. Which one is better? We don't know. The thing is that when we have these more complex materials, we have a lot of noise. How do I denoise my data? I don't know. I know it's possible, but I don't know how to do it. Because to really be able to analyze this signal that is extremely weak, but holds a huge amount of information, I have first to denoise, to take out everything that's not interesting for me. Then there is another question that I ask. Okay, I'm looking at this energy range. Is really at this energy range that my most important signal is? I don't know. But the information that I collected on my experiment is there. But there is no human power able to analyze all the data. And then, at the end, there is also another explanation. Remember that I told you that we had also for the protein that was inhibited. We got, when we do our analytical calculations, the line width as a function of Q squared, the same way that I got for the dental cement. So if we use some theory, and some of you that listen to Gerhard Kneller talk, that's what he was explaining. That what happens here is that when we inhibit the protein, we change the potential. So, and this is the kind of information that I really want to get, is on the potential. Because if I have information on the potential, I have information on my single particle. And this is a very new publication also. It's a very interesting and very challenging publication that came out because of the first XFEL experiments, where we can really look at single particles, where Abbas says single particle techniques and the role of structural viability in biological function. And the point here that makes us think is that when we are looking at dynamics, I'm looking at how things are moving fast. When we talk about the structure, it's a snapshot. How do you tell me that that structure is the correct one? That structure is the correct one that you measure in that particular time. But in things that are very flexible, they will change again. So I just have an average information. And that is what we want to get at the end of the day. These potentials, how wide they are, and how noisy they are. So this is the conclusion that we got from our workshop. I took it from a slide that John presented. And machine learning can indeed help us modifying and changing and revolutionizing the way that we do analysis of data that come from the large scale facilities. Because here we need a lot to understand what we are measuring, not just using some analytical calculations, but really having some theory behind it. We need help for automation because we need to find new ways to configure our instruments and to do setups. How do we really analyze this data that comes very fast on the flow? How do we decide to say, okay, next, stop, go, your error bar is big, but we can denoise your data, we can move forward. And then automated discovery. How can we really do better? And I think in the elastic neutron scattering experiment that I showed to you and the analysis is really a very interesting example. Because this type of data, we have a lot of them that we can use for training. And what we can do is that we have very small, the molecule we know. So we just have to figure out how this molecule is configured in this crystal. And then it is indeed easy to underestimate the complexity. It's not easy, right? It's not easy the problem we have. It's not easy the problem I'm putting forward. However, developing collaborations and having these opportunities to talk to each other is very important because we need to develop a common language. I know for me also it's not very easy when I start an experiment with an epidemiologist or I start an experiment with someone in odontology or someone in chemistry. It's a language development and we have to learn how to understand each other's language. Well, at least I hope that not very far from now I will understand really the difference between AI, ML, and DM. And then I just would like to thank all these people that participate on the first workshop that we had. I would really like to thank Christina. She's not here because it was Christina, I met her last year and we started discussing with Christina is that I got really the opportunity to come and talk to you. Thank you very much for your attention.
Just one comment relating to deep learning and machine learning in general. I have used unsupervised techniques, so techniques where you don't train against some data, but you simply more because there's something that is different from the rest, I think you should really look into unsupervised techniques. At least that's my experience. Also because you don't need to generate labeled data before you experiment, so you're not biasing, you can say, your search in that sense. And the other comment I just want to say is, because I saw your slide and I think there's this push to make it high throughput or what would you say, like fast. And I just want to say that when the little machine learning I do, it takes longer often than more simple analysis. So I understand you want automation, it has to be fast, but if you do label, you need to look at bias variance trade-offs. So like, are you overfitting your data? Are you fitting noise? Are you making too simple a network, etc., etc.? So just take into consideration that I think it's promising, but it's also more complicated. So sometimes even if it sounds like it's more efficient, it can be very time-consuming to get it to work.
But molecular dynamics is the same.
Yes.
I need much longer time in doing molecular dynamics than to do my experiment. But sometimes it's the only way that I can use.
No. It's absolutely. It's just that people present it as this thing like, okay, we're gonna do neural networks.
No, you're not.
And, someone's going to sweat over it for a long time.
So I think related to that, I think there are two different sort of domains. One is if you want to improve on a physical model, so you have a physical model, but it's not accurate enough. That's sort of one setting, and there you might have to pay a large price. And the other setting is that you have no physical model, and so you don't know at all how to model it, and that whether you can, and that you have a lot of data, and that you can try to train a model where a flat box model, and those are quite different, I think, domains. So in which of these cases do you actually have physical models, and is it often a problem that they're not accurate enough, or is it more a problem that you don't know at all how to model it?
Well, as I showed to you in the molecular dynamics case, in the magnetism, you have the model, right?
Yes.
So
in that case, is molecular dynamics accurate enough, or is it just too slow? Do you need something faster, or is it not accurate enough, and do you need to go to quantum sort of theory in order to get the right results out of the simulation? And is that
I think I asked John to answer this question for you.
Sometimes you know the model, sometimes you don't.
Right.
Sometimes the model fits very well, and sometimes it doesn't. When it doesn't fit, that's the interesting physics or science.
Right.
Because the models are typically straightforward. For DFT, it's a mixed bag. Because of the complex, depends what you're measuring. If you're measuring some simple, calculating some simple structure which works well with DFT, that's okay. But then you run into real world situations where you have to impose certain precursor assumptions about the system to make the calculation converge, which is pretty much the most simulator or modelling the interaction between electrons to get around the many body problem, which you can't solve in any way. Right.
And I think here, if we go back to G's, that is the same thing with my dental cement hydration, right? There was no way that a model that we have in mind can fit these points, right? So that is why, in this case, this colleague of mine had to go to some theoretical analysis, and we went to molecular dynamics.
Right. Because there's, I think in machine learning, there's some developments now that people try to either sort of wrap physical models with machine learning to target correct for errors, or the other thing is that you can try to approximate DFT calculations by, if you have lots of data, right, you can make an effective force field for that particular system.
So here we can go back to this one. Normally, when on DFT, what you could also do is that you can do the structure again to model to your scattering, to your inelastic data, right? And there are some people that have done that. So you optimize your structure again to get your DFT optimized till you get it, right? So here it is a very good example because, again, we have this problem with two structures that are very similar, right? That were solved by crystallography. But again, it doesn't fit my model, and it still kind of doesn't fit my model. So again, what we are talking about are molecules that are flexible. And when you do the structure, you consider normally low temperature because then everything is stopped. So I don't know my structure. In reality, I don't know the structure of paracetamol that I take. I kind of. But you see, it's not, I'm not making claims that it's wrong. I'm just making claims that if, when I do a calculation, my data does not fit, I have a colleague that always said data don't lie. So yes, okay, I'm happy with what I'll have, but I can do better.
Okay, any other comments or questions? I want to, could you say a little bit more about this, what was it called, this SGDML or whatever? Do you have any rough idea of what they do there? What's happening?
Well, the rough idea that I know what they do is that here, they basically, they consider local symmetries.
Okay.
Right, so basically, for what I understood, but I may be wrong, what they do is exactly what we did by brute force. So you have a molecule, and you don't put that molecule in a real structure, as you do on GFT. But you take that local symmetry and use that in different ways. That's how I understood it.
Yeah, I was just wondering what this gradient domain, what this refers to. But
Yeah.
Yeah, I just looked it up. I found the GDML paper that's before the SGDML paper. And basically what they say is instead of, I guess I'm not sure what this P is, but instead of computing gradients explicitly, they're doing some kernel ridge regression to compute gradients. So they're trying to get calculate gradients.
all right. I think it's time to stop, but there's coffee and cookies here, and which will be brought onto the table. So please stay for a chat and have a cup of coffee afterwards. And thank